on to our next talk, nicely linking in from Dr. Tappi. Silvia is in the Department of Agriculture and Food Sciences at the University of Bologna, and she will present to us about physical and structural characterization of creamed honey obtained by a guided crystallization process. Silvia obtained her PhD in food science and biotechnology in 2016 at the University of Bologna, and now is a junior professor at the Department of Agriculture and Food Sciences at the very same university. Her research is focused on food processing optimization for increasing food quality, sustainability, and innovation. In particular, she investigates emerging non-thermal treatments for food formulation, processing, and stabilization, and the physical and structural properties of both vegetable and animal-based products. She participated in different national and international funded research projects and presented her work in many international conferences. She has co-authored and authored more than 40 scientific publications in internationally recognized journals, and she holds 1 patent. Over to you, Silvia.
Hello. Hello, thank you very much for the introduction. I'm very happy to be here. So I'm sharing my screen. Okay, so thanks, Daniel, for the kind introduction. And I'm very happy to be here to talk about this topic. And I would also like to thank Sandra for the interesting presentation that she introduced many concepts about honey crystallization that will help to better understand what I'm talking about. My presentation is related to a specific type of crystallization, so guided crystallization, to obtain a product known as creamed honey, which is a product that is started to be known among honey producers and consumers, but is still not known by many consumers, particularly here in Italy, where I am. So I'm Silvia Tappi, I'm from the University of Bologna, and what I'm presenting today is related to an investigation that we carried out on the physical and structural characterization of this product, creamed honey, obtained by guided crystallization carried out in different ways. So first of all, as we've seen from Sandra's presentation, crystallization of honey is a natural process that occurs in all honey due to the fact that honey is a supersaturated solution, and so it's generally unstable. And the sugar that crystallizes, as she said, is glucose, and so it turns from a liquid to a solid state and create this granular composition that is perceptible by our senses. Crystallization, from a commercial point of view, is generally considered a defect because the increasing phase of crystals promotes some changes in the textural rheological properties that affect consumer acceptance. Moreover, when crystallization is spontaneous, sometimes can lead to an homogeneous product, depending, as Sandra explained, on the composition of honey and of the storage condition. But it can be characterized by the presence of very coarse crystals that can often produce these problems that you can see here in the images, so retraction spots, incomplete crystallization, the occurrence of the typical phase separation that is very easy to see in commercial honeys. So when we have the glucose, crystallized glucose at the bottom and the watery phase on top that contain mostly fructose, and also the increase in water activity that can lead to honey fermentation by osmophilic yeast. It's not a problem for food safety, but of course is a loss of quality. So generally, industry tends to reduce or avoid or delay the occurrence of crystallization. One method is, of course, pasteurization. That, however, as you know, reduces the quality of the product. Another strategy is to guide the crystallization. So to actually promote the occurrence of crystallization and to complete the process before trading the product in order to obtain a product that is uniform and stable during storage. This technique has been first patented by Dice in 1935 And it's involved the using of a crystal seed. So the addition of a mass of crystallized honey, they would act as to initiate the process of crystallization. So as a primary crystallization nuclei. This method, according to this pattern, allows to obtain honey that is characterized by specific rheological and textural properties. So it's soft, spreadable, is very smooth texture, and a lighter color. Is generally known as creamed honey. However, the consistency of the final product depends on the composition of the initial honey, as Sandra explained. So, but however, it allows to avoid the crystallization defects and to obtain honey which have a homogeneous characteristic. These are the steps indicated by the patent. As you can see, there was a pasteurization step to remove yeast and then the further cooling. And then the most important step is the addition of the crystal nuclei. So what is this crystal seed? Is granulated honey, crystallized honey, that is finely crushed in a grinder in order to obtain very fine crystals. And the patent suggests to add it in a percentage of 5% compared to the mass of the liquid honey, and then give indication on how to mix it in order to thoroughly distribute the crystals in the mass. And then a storage at 14 degrees that we've seen previously is a temperature ideal for crystallization, so at which crystallization occurs faster. In the more innovative processing, the pasteurization steps is generally omitted in order to avoid quality loss. And we define this in our investigation as a static method. Why? Because the honey, after mixing, is stored in a static way, so in glass jars during the crystallization. The method indicates actually a timing, so 34 hours to reach full crystallization. However, this time strongly depends on the composition of the honey, so on the crystallization trend. So it's not possible to actually define a time that is necessary for full crystallization for all honeys. More recently, producers have introduced the mixing of the mass in what we call the dynamic method. So after the addition of the crystal, the mass is mixed, is stirred. Some producers actually just stir the mass at irregular intervals. We wanted to standardize this mixing, and we used an in-house built prototype. That is the picture here is a lab scale prototype with a small volume of just 1.5 liters. And inside the chamber, there is kept at constant temperature. There is a propeller that is rotating constantly at 14 RPM. So after the crystal addition and the mixing, the mass is kept stored at 14 degrees, but under constant mixing and movement. As a crystal seed, we used crystal citrus honey that was finally crushed with a grinder, and we added this to 5% to different honey samples. In order to understand the influence of the composition of the honey, we selected some honey and we mixed them in order to obtain specific compositional parameters. As you can see here, we tried to standardize water content to have more or less the same amount, so to have that as a constant. And then we obtained different fructose-glucose ratio. Alexandra explained very well before, it is related to the crystallization trend or the crystallization rate. Now here we are adding already crystallized mass that would act as initiation. So it will increase the crystallization trend, but these samples are characterized by different fructose-glucose ratios, so we define them as fast, medium, and slow crystallization rate, and we call them FMSC. And then so basically we had 3 samples of honey, we added 5% of crystal seed, we mixed them, and we subjected to storage in a static condition, so in simple glass jars, or dynamic condition with this prototype, so under constant mixing. We decided to characterize the samples during crystallization to evaluate the crystallization kinetic, but also the textural, rheological, color, and structural properties of this product. So we used as a technique to evaluate the crystallization, differential scanning calorimetry. I don't know if any of you is confident with this technique, but this technique allowed to evaluate the amount of crystals present in the mass, exploiting the fact that glucose crystals generate heat when they're melting. And this is a typical melting thermogram of crystal of glucose that melts between 30 and 50 degrees. And this is the evolution of the amount of crystals in the different honey types subjected to static crystallization. So here we have days on the x-axis, and you can see there is an increase of the amount of crystals with time. And the fast crystallization glucose, the samples, the blue one, the one characterized by the low FG ratio, fructose-glucose ratio, had a faster crystallization. So we confirmed this, this fact. At the faster crystallization, so glucose was crystallized faster, but also reached a higher amount of crystals. Okay, so the crystallization is faster, but also there is a higher amount of glucose is crystallized. This was the time necessary to reach full crystallization. So 20, 90, 102 days. After that, there was no more increase in the crystals. What happened when the same honey samples were subjected to constant stirring? This is what happened. So the crystallization occurred, but in a faster way. So we still had the difference between the three honey. So they still had the crystallization rate proportional to the fructose-glucose ratio, but the time necessary to obtain full crystallization were 11, 16, and 32 days. And please note that the amount of glucose crystals were the same for each sample. So the crystallization occurs 2 to 5 times faster when the mass was kept under constant stirring. But what about the structural characteristics? So we evaluated the texture of the product with a penetration test. So the probe penetrating into the sample for 7 millimeters. This is a method that we find in the literature. And this is a texture profile of force versus time obtained by the different samples during storage. We consider the highest peak force as hardness of the sample, and we consider the negative area as adhesivity. And here we can see, maybe, yes, the hardness and adhesivity of the samples subjected to static crystallization. So we can see that there is an increase of hardness, that the behavior is similar to the crystallization rate. So the fast crystallization sample reached a higher amount of crystallized mass and a higher hardness. And this, as you can see, was proportional in the three samples, as we've seen for the crystallization rate. And this is due to the increased crystalline phase that is forming a solid structure where the crystal create a matrix and that have a cohesion in the matrix that increase with time. What happened when a sample were subjected to dynamic, so constant stirring? The samples are actually present in the same graph here, but we cannot see them because the values they reach are very low. So I make larger these areas. So we can see that the textural parameters were remarkably different. So considering only the blue sample, so the fast crystallization sample, as you can see here, our full crystallization, he had a hardness of about 50 Newton with the static method. In the dynamic method, the maximum hardness reached was 0.04 Newton. So the difference is quite remarkable. The other two samples actually kept the same hardness and adhesivity during all crystallization, during all the crystallization process. So this parameter did not change, although the crystallization reached its maximum, so reached the same amount of crystallized mass. So we observe remarkable differences in the textural parameters. We also evaluated the rheological behavior of the samples by measuring. Here we used a plate system in a steady state condition. And here you can see the graph for the dependency of shear stress and shear rate. And the blue sample here at the bottom is a liquid honey that shows a Newtonian behavior as according to literature. And as you can see, the sample is moving upwards as the crystallization time increases. And what we observe is the appearance of an hysteresis loop that started to appear, describing a thixotrophic effect that increases with increased crystallization time. This behavior has been already observed in the literature and is associated with the destruction of the crystalline agglomerate. And so the crystallized honey is a non-Newtonian fluid, again, in accordance with the literature. Here we can see what happens during the dynamic crystallization. So again, as you can see, there is a moving upwards during crystallization time, but the hysteresis is not appearing. And actually, the crystallized honey in the dynamic method allowed to maintain the Newtonian behavior of the honey. Here we can see the viscosity calculated of the honey samples. So as you can see here, the initial value of the sample were exactly the same for the 3 samples, and they increased during crystallization as expected. But if you look at the difference between static and dynamic method, again, we can see a striking difference. So the dynamic method allowed to obtain significantly lower viscosity compared to the static method. So considering the results that we've seen before, so both methods led to the same amount of crystallized glucose. However, the structural characteristics, the rheological and textural characteristics were very different. And we attributed this to the size of the crystals, because the rheological behavior depends on both the amount of solid phase, but also on the morphology of the solid phase. So we wanted to evaluate the size of the crystals. We use an optical microscope with a polarized light. With this technique, we can visualize crystals. So in a completely liquid sample, we can see just a black background. This is how appear the samples after the addition of crystal seed. So we can see the black background, and we can visualize the sample, the crystals that we added. So this is our crystallization initiator, the crystal seeds. This is the appearance of the sample crystallized with a static method. So we can see that as a number of very big crystals that reach a dimension of 200, 300 microns, and a large crystalline structure. And this is how the honey appeared after full crystallization with the dynamic method. It's different from both the previous one. First, we can still see these crystals that we believe are the same crystal that we added, so our crystal seed, but then we don't see anymore the black background. We see a gray background dotted. This indicates the presence of a huge number of very small crystals or microcrystals that have dimension less than 10 microns. So crystals that are not perceptible by a human mouth, and so they can give this creamy texture. And also we cannot see the big crystalline structure that is formed during the static method. So we were able to determine the fact that the continuous stirring, the constant movement of the mass, basically inhibited the growth in size of the crystal. So what happened during static method, the crystal grow inside and become large, while the constant movement inhibited this growth, but favor the appearance of a large number of very small crystals. The size of the crystals, apart from affecting the textural and rheological parameters, also affect the visual quality of the sample, because crystals of different size have different refractive index. And this is the appearance of the liquid honey after the addition of the crystal seed. So at 5%, it was not possible to see just with the eyes. And this is how the same honey sample appeared after crystallization with a static mode. So you can see it changed the appearance due to the formation of this crystal mass. This is the same honey sample crystallized with a dynamic method. So as you can see, it's different from both the previous one. It's very white and glossy. And this is due to the difference in the crystal size. So finally, I want to show you the principal component analysis. For who is not confident with this type of technique, is a statistical technique that allows to simplify data of multivariate analysis. And it consists of a vectorial space in which there are parameters and samples distributed. And basically, as much far away they are the sample from the other, as much different they are. So we can see here that on the x-axis, we have the parameters related to this rheology and texture. And on the y-axis, we have mainly colorimetric parameters, so luminosity and red index, they are the opposite. And here we have the sample at the beginning, so the liquid samples, where we can see that the fast crystallization sample was different compared to the others, mainly for color. They are characterized by a different color because there was different mixture of different type of bonies. But they were characterized by the same rheological parameters. So they are on the same side of the x-axis. What happened after crystallization? As we can see here, samples moved far away from the liquid samples. So the farther away they are, the more different. So we can see the fast crystallization honey is the one that was subjected to the highest change. So it was characterized by higher crystallized mass, a higher increase in viscosity and hardness. So the faster crystallization trend led to higher changes during crystallization compared to the others. This using the static method. What happened with the dynamic method? So keeping the honey constantly stirring. These are all the sample at the end of the crystallization. What we can see is that first, they did not change so much as texture and rheology parameters as we've seen before. So they are basically on the same level on the x-axis. They just move downwards. They move downward where the L parameter is found, so the lightness, and far away from the red parameters, so they become more white and glossy. But another important thing that we've seen is that why the different honey samples crystallized with a static method were far apart from each other, these three samples are very close together. So it means that they are very similar to each other for all the parameters investigated. So in conclusion, guided crystallization is a good strategy to avoid the crystallization defects, and also can be used to obtain a honey with peculiar characteristics, so kind of a product innovation. It's a product that is mostly known for what I know in the US and less in other countries. With the traditional static method, the products obtained have characteristics that depend strongly on the composition of the initial honey. While on the other side, the dynamic method allows to obtain a product that is characterized by smooth and creamy texture with all the tested honey types. So we cannot really talk for all the honey types, but in the range that we investigated, they are characterized by smooth and creamy texture, but in particular similar and homogeneous characteristics, also in terms of visual quality. It's a product that is stable during storage because it's fully crystallized, and is characterized by peculiar characteristics. So as we've seen, the texture is particular, the color is particular, so it's different from both the liquid and the traditionally crystallized honey. So it might need some specific marketing strategy, but we believe that is a very interesting product that should be further investigated. So I thank you for your attention. And
thank you,
okay.
Silvia.
I have to interrupt. Okay.
Thank you, Silvia. I got a couple of questions. Number one has been sent to me directly. Question of scale up. How easy is it to scale up your technology?
It's actually very easy because I mean, what I showed is a built prototype of lab scale, so 1.5 liter, but in reality it's quite simple because it's a large a large sample, large container. Inside it has a propeller that is constantly moved. There are some larger, I think that the larger prototype that I've seen carry 50 liters of honey, but I think that the scaling up is quite easy.
Okay. Jahinder's asking, well, first of all, he says compliments to your presentation. He's asking, have you looked at supercooling the liquid honey and injecting the crystallization points like they do in ice cream production?
Oh, so have you looked at supercooling the liquid honey? No, actually, no. This is something that we haven't done. I'm not sure what could happen, to be honest. I'm thinking, but it could be an interesting, again, another interesting opportunity. But
yeah,
okay.
I mean, I know Jahinder, he's a rheologist, so I guess he has a private interest in that. Ayumida's asking, undoubtedly crystallization alters the organoleptic properties of honey. How about the nutritional composition? Is it also being affected by crystallization? Does your work focus on this?
Well, for what I know, the crystallization should not alter the nutritional composition. I mean, it's simply the passage from liquid state to solid state of the glucose. The glucose in solution goes into glucose crystal. So every other parameter should stay the same. What changes is the water activity of the sample. So during crystallization, water activity tends to increase, and this might lead to problems of fermentation, and that in turn can for sure change the nutritional composition. But we haven't investigated this. So, but yeah, I think the main problem is related to the occurrence of fermentation, because the crystallization itself does not change the nutritional properties, for what I know anyway.
Okay, I got another question in. I think it's more or less on the same. In the same direction. Did you continue to work to look towards mouthfeel and flavor delivery of dynamic method versus static?
No, no, in the sense that what we are trying to do, what we started to do was some specific and focused sensory analysis. It is not my expertise, but we were working with a team that investigated a specific type of sensory analysis. I think they were mainly investigating the textural properties, but of course the change in structure can alter the flavor perception. So that's for sure is interesting to evaluate, also to see if there is a change or there is an homogenization of, yeah, that's an interesting aspect.
Okay. Okay, what is the last question? The last speaker spoke on adulteration and crystallization, which makes crystallization seems not too good for any honey consumption, but you are presenting it as a good state for honey or of honey. Please, would you say crystallization of honey is good? Philosophical.
Okay, so crystallization is not bad in itself. It's bad from a commercial point of view, because of course the honey, also the fact that crystallization can occur in a homogeneous way. So when it's not controlled, so as we've seen, it can have retraction spots, you can have the separation of phase, so it's a quality loss. Is not bad from a nutritional point of view, as I said, unless there is fermentation, which can occur. But on itself, the crystallized honey does not have different nutritional property or quality properties. I investigated as a strategy to obtain a stable product, homogeneous, and also to have a product with some specific sensorial characteristics that might be or not appreciated by consumers. I guess it depends on taste. So is good or bad? I don't think it is neither good or bad. Of course, it depends how you look at it. But from a commercial point of view, the uncontrolled crystallization is definitely a bad thing from a practical point of view.
Okay, there's one last question. Have you ever looked into heather honey and its thixotropic properties? Correct?
No. No, this is a honey. No, this is with. I never work with this particular honey, so I don't know if it has some specific rheological properties. So no, I haven't.
Okay. Thank you, Silvia. We move on to our